all also they are also fungi are also now saprophytic or parasitism. But what type of nutrients are present in fungi? They are all the they are not to prepare food material, but how to the food material to importance of the food and food processing in this one. Fungi lack of chlorophylls are present. Therefore, they can't synthesize their own food material. That's why this is called the heterotrichous algae, not autotrophous. These are the heterotrophous fungi. Yes, why they are not to prepare food own. That's why their lack of chloroplast is present. That's why they are the heterotrophous and they not, do not to prepare own food material. That's where the chlorophyll is absent. That's why they are the heterophilous. And they can't, next one, for depending on for where and how they get, how and where they and how they get nutrition. Fungi are of following types. Yes, they do not to prepare the food material whole, no. That's why the fungi divisions are two types of fungus. One is the saprophytic, another one is the parasitism. One is the saprophytic and another one is saprophytic. What is the saprophytic and parasitism? Saprophytic fungi, they obtain food from dead and decaying of the organic matter. They obtained from where you can observe to the saprophytic fungi. They obtain food from dead bodies and decaying organic matter. They create digesting enzymes to outside which digest the substratum and then absorbs nutrients. The best example for saprophytic fungi, Mucor, Agaricus, Rhizopus. This is Zygomycotina family members. This is a saprophytic. Okay, here this the fungal. They do not to prepare the own food material. That's way depending of the get nutrition different ways. That way is two types. One is a saprophytic parasitism. Saprophytic meaning obtain food from dead bodies or dead decaying organic matters. These create the digestive enzymes. These outside the digest the substratum and absorbs. Yesterday I will explain hyphae, substratum like this. Okay. This best example saprophytic, Mucor, Agaricus, and Rhizopus. Coming to the second one. What is the second thing? Parasitism. What is the second one? Parasitism. They obtained food from living organism. Obtained food from living organ. Saprophytic. What is the saprophytic? Food from the dead and organic matter, decaying organic matters. But parasiting, they obtained food from living organisms. They may be facultative or obligate. Very important. They may be facultative or obligate. Facultative parasitism or obligative parasitic. Both are present in the parasite. First one, obligate. See in the picture also, you can observe. This is a saprophytic picture. You can observe to the saprophytic picture. Coming to this one is a parasiting fungi. Ladybug. This one is a ladybug. These facultative parasitic grow on a variety of different types of tissues. For example, Eustilago. Eustilago is the first example. This is a facultative. The second one, obligative parasiting, grow upon suitable host. Suitable, any host uh, growly upon the suitable host. Obligative. The best example obligative downy mildew diseases. Downy mildew diseases. Coming to the parasitic fungi that grow on surface of host cell and absorbs food through the hostoria are called ectoparasitism. What is that one? Some parasitic fungi some parasitic fungi that grow on surface on the host cell absorbs food material through the hostoria are called ectoparasitism or ectophytic parasite. Example, mucar and Ersipia. When parasitic fungi grow inside the host tissue are called endoparasites or endophytic parasites. Example, Puccinia and Pythium. This is the second parasitism. Parasitism you can observe to the two. After that one, third one, predaceous. Predaceous fungi. Predaceous type of fungi some soil fungi. This is called the soil fungi. Develop ring-like noses to trap nematodes. Annelidas and nematodes. See, you can picture, you can observe the picture. This fungi, soil fungi, develop ring-like nose. See, this one ring-like nose to trap annelidas, nematodes. Best example, Arthrobotrys, Juphagus, Dactyella, etc. Okay. Coming to the fourth one, symbiotic, relationship, lichens. They live in mutualistic relationship with another organism by which both are benefited. The two common examples are lichens and mycorrhiza. What are the example? Best example, mycorrhiza. The second one is lichens. Lichens are symbiotic association between the algae and fungi. The fungal partner is a member of Ascomycetes or Basidiomycetes. Different classes are there. In the lichens, you can observe to the both two. One is the Asco, second one is the Basidiomycetes. That provides water and nutrients, while the algae partner is a green alga or cyanobacteria. Chlorophyll is a green algae. Cyanobacteria is a blue-green algae that prepare the food by photosynthesis. That food to absorb the fungal Aspergillosia. They both are beneficial are there. Some algae is not to grow in soil. Yes or no? But like fungi is grow on the soil also, in the soil also. But these both are too beneficial. That is a symbiotic. Coming to the mycorrhiza. Here, mycorrhiza or mycorrhizae are the mutualistic symbiotic association between soil fungi and the roots of most plant species. Very important. What is the main in this one? Mycorrhiza or the mutualistic symbiotic association between the soil fungi and the roots of most plant species. According to the carbohydrate theory, Bochcom scientist in 1949, the plants that grow in soil deficient in phosphorus, nitrogen. The plant grow in the deficiency of minerals, phosphates and nitrogen. Nitrogen and high intensity light developed mycorrhizas. Okay, next one, the most common type of mycorrhiza are the ectomycorrhizas, ECM. What is that one? Ectomycorrhizas. Mycorrhiza, that is the ectomycorrhizas, ECM. And the endomycorrhizas, and also as arbuscular mycorrhiza. This is called arbusculus mycorrhiza. The two groups are differentiated by the fact that the hyphae of ectomycorrhizal fungi do not penetrate the cell wall of the plant root cell, while the hyphae of arbuscular mycorrhizal fungi penetrate the cell wall. This is the mycorrhiza. See, you can observe lichens with, this is the one lichen. And this part is the internal structure of the lichen. See, this layer, green color layer is there, no? In that green color layer is called algal layer. And down part, it is all or eye face. Both are attachment structure. This is called the lichens. You can observe internal like this, external like this. Okay. Next one, plant showing with and without mycorrhiza. With mycorrhiza, this one. This one is without mycorrhizae. And coming to the, what is heterothalism? What is homothalism? Very important. Heterothalism and homothalism also most important in fungi. The Blackman scientist uh, Black Sea. The scientist name is called the Black Sea. This Black Sea scientist in 1904 discovered matting type of the genetical distinct strains in mucars. See, heterothalism is the homothalism is. He called and in the Black Sea to explain in heterothalism, example for the example mucar. Okay, he called fungi with different matting types are called heterothalism. Different mating type. Different mating types are called the heterothalic. Heterothalic and fungi. Without mating type are homothalic. This is the different mating type or heterothalism. Without mating type are called homothalism. You can observe both the pictures. Hetero, this one. This is the one fungi. After meiosis to form this one. And these two undergo the mitosis division. These two mating with, both two mating with like after meiosis again to form. This is the heterothalism. Homothalism, see, here both combination to undergoes meiosis to after that one to form only one. After mitosis and mating type is switching, the both mating to after that one, this is the homothalism. Nowadays, we call some fungi and algae homothalic. The that is the I'll explain in the before lichens. Okay, here nowadays the scientists to explain some fungi and algae all are the homothalic. If both male and female gametes produced in the same individuals can fertilize each other, and heterothalic if the gametes can only be fertilized by the gametes gametes from another individual of the same species. This way, this is called the heterothallism introduced variation in the species. You can observe this part, this one. Okay. Coming to the next, reproduction in fungi. What type of reproductions are there? How many reproductive structures are there? And what is the main source of the reproduction in fungi? We will discuss now. Okay, coming to the reproduction. In fungi, Hmm. In fungi, reproduction may takes place by three methods. You know what is the three methods are there? One is the vegetative reproduction, second one asexual reproduction, and third one is the sexual reproduction. These all types of reproduction you can observe in the where in the fungi. Okay, reproduction, the first one, during asexual reproduction, sexual reproduction in fungi, asexual reproduction in fungi. Fungi reproduction, three types, both as, but mainly asexual and sexual. In some fungi, for example, fungi imperfecti. Some I'll explain to the some example. Imperfecti fungi, that is called Deuteromycotina fungi, reproduced is by asexual method only. In the Deuteromycotina, you can't see asexual reproduction. Here only you can observe to the asexual reproduction only. In some other, it is pathogenetic. But in majority of fungi, reproduction occurs in two well-marked phases. That is the asexual, imperfecti. Sexual is the perfect. Imperfecti and perfect. Differing from the sexual reproduction is parasexuality, demonstrated by Ponto Corvo in scientist in 1954 in the member of fungi imperfecti. It has since been reported in other group of fungi as well. As asexual reproduction is being used here in broader sense, including vegetative reproduction also. Okay, usually the term vegetative reproduction is used for the development of new individuals from unspecialized detached portion of the thallus. This is the in that one is the reproduced by the vegetative. Okay, in asexual reproduction, the new individuals arise from specialized reproductive bodies produced by the thallus without involving any nuclear fusions. However, since biological, there is no difference in the vegetative and asexual reproduction, both being non-sexually. They are put together under asexual reproduction. Here, asexual and sexual reproduction process, main spores are the essential. Both are the spores. Spores formed after meiosis division. That spores name is called meiospores. Okay, in the meiospores, but best example, see, this is the vegetative reproduction mode. This one is the sexual reproduction mode. In the spores, what type of spores are there? Ascospore, basidiospore, and sporangiospore. And those resulting from mitosis called mitospores. Meiosis spores, meiospore. Mitosis spore, mitospores. Called mitospore. Example, mitospore, zoospore, aplanospore, conidia, and uridospores. This one is the diploid body produced as a result of sexual fusion is known as zygote. Within the lower fungi, the term as resting spore. Zygote is called resting spore, oospore, or zygospore. In higher plant of fungi, the zygote is represented by the diploid nucleus. This diploid nucleus produced in a cell that name is ascus or basidium. This diploid nucleus, after undergoing meiosis, result in the formation of haploid nuclei, serving as center for the haploid sexual spores, that is called ascospores or basidiospores. This is a best example for three types of. Coming to the first reproduction is the vegetative reproduction. Vegetative reproduction, this type, the first method is the vegetative. This type of reproduction, a part of mycelium separate and form a new individuals. New individuals. The various methods of vegetative reproduction are, that one is the fragmentation method 1, fission method 1. Next one is the budding method. First one is fragmentation. What is the fragmentation is there? Here, main method, the hyphae break into small pieces or small fragments. Each piece, upon getting suitable condition germinates to form a new mycelium. A new mycelium. This is the fragmentation method. Coming to the fission method. Fission is, yeah, sorry for the disturbance. Now I am sharing. Sorry for the disturbance. Yeah. Yeah. This is the fragmentation. The hyphae beak. The hyphae beak into small fragments. Each pieces upon getting suitable condition when that germinate new mycelium or new fungi plant. This is the first one. The coming to the second one, fission. This method involves the splitting of cell. Fusion is the splitting of cell. You can observe the fission methods. This one is a fission method. Observe to the picture. Fission method. This method involves the splitting of cells into two daughter cell by the formation of a construction followed by cell wall formation. This is the fusion method. The splitting here, you can observe the splitting of the cell into a two daughter cell by the formation of a construction followed by a cell wall formation. This is the fusion. Next one is the budding. Okay. Next one is the budding. Budding of vegetative fusion is the this fusion characteristic. First one is the fragmentation. Here, detached fragments of the hyphae, suitable condition gives rise to a new individual. This hyphae, this happen frequently in nature and is the method used for cultivation of fungi in the laboratory condition. Keep in your mind, this is the most important. Okay. Coming to the fission, this is the characteristic of a bacteria. You observe the when bacteria cells, this type of fusions are there. Bacteria and occur only in the fusion yeast cell also. Best example yeast. You can observe the picture also. Yeast. That yeast name is called Saccharomyces yeast. In fusion, the cell divides in transfer plane into two cells by construction and later formation of a cell wall in between. Coming to the budding. Budding of here, a small bud formed from the parent cell, which gradually increase in size, receive a part of nucleus, and the cell wall formed with a separate the daughter cell from the parent cell. The budding of vegetative cell common in yeast. This one also very common in yeast. In yeast, a soft zone appear on the cell wall. You can observe the very soft zone upper in the cell wall, upper into the soft zone upper on the cell wall, which bulges out, constricts, and finally pinches off to form a daughter cells. See, daughter mother cell to form the daughter cells. Mm, Budding may occur on spores also. On spores also. These three are the next one. Sclerotia. Sclerotia. These are the, see, you can observe the sclerotia, this part, the picture. This is a penetrating bodies formed by the compact mass of intervone hyphae. What is a sclerotia? Penetrating bodies. Penetrating bodies formed by the compact mass of interwoven hyphae. This sclerotia under suitable condition germinate to from new individual. Claviceps purpurea sclerotina. Claviceps purpurea is the in the laboratory condition develop the fungal is the claviceps purpurea, and next one is the sclerotinia. Okay, next coming to the another one, vegetative reproduction, rhizomorphous. What is the next one? Rhizomorphous. Rhizomorphous, there are root-like elongated. You can observe the picture. All root-like pictures are present in this one. Root-like elongation mycelial strands are there. They remain dormant under unfavorable condition. Under unfavorable condition, they develop into a new mycelium plant. Okay. These all are vegetative reproduction. How many? Fragmentation, fusion method, budding, sclerotia, rhizomorpha. These are all to the vegetative reproduction. Coming to the asexual reproduction. Here, main asexual reproduction, commonly you can observe to the spores. Different types of spores to observe. It commonly takes place through spores. Takes place through spores. These spores, takes place spores. Either the spores is the either the spores either Either spores, either your spores main important motile spores or non-motile. You know, flagella are present, non-flagellas are present. These spores are the flagellas or non-flagellas present in a specialized part. That part is the various types of spores are present. In this one, the first one, spores main important are the most common method of asexual reproduction in fungi. The term spore is used for any small propagative, reproductive, or survival unit which separate from a hyphae or a sporogenous cells and gives rise to a new individual. In contrast to the vegetative mycelium, the spore is characterized by first one, cessation of cytoplasmic movement. Second one, small water contact. Third one, slow metabolism. And fourth one, lack of vacuoles. In mycology, study of fungi, spore is a general term capable of wide interruption. It is qualified by prefix to indicate the particular type. These may be produced asexual or sexual. Thick-walled zoospores or oospores, ascospores, basidiospores, or spores formed as a result of sexual reproduction. These are called meiospores. These are called meiospores. Asexual spores are called mitospores. Asexual reproduction spores are called mitospores. Asexual and sexual reproduction is the meiospores. Asexual spores is the mitospores. Asexual reproduction spores are two main type. But the two asexual spores are also are of two main type. One is the sporangiospores and conidia. What are the two? One is the sporangiospore and conidia. Sporangiospores are formed within a sac-like structure called sporangium. Plural sporangia. Plural is the sporangia. See, here I'll explain the first one, sporangiospore. Next, I'll explain to the zoospores. This is the sporangia. Sporangia, the sporangia sac-like structures are, see, this is the sporangiospore. Asexual spores are two main sporangiospores in the, and the second one is the conidia. Conidia one, sporangiospore one. Sporangia spores, you can observe the first one. Sporangiospores are formed within a sac-like structure. See, this is a within a sac-like structure called sporangium. That is the sporangium which is born on undifferentiated or specialized hyphae structure. See, undifferentiated and specialized hyphal structure called sporangiophores. That is a sporangiospores. These sporangiospores are aplano or non-motile spores. Lack of flagella. Flagella is lack or absent. Formed inside the sporangia. Example, rhizopus, mucor. These spores may be uninucleate to multinucleate and possess two-layered cell wall are present. Coming to the conidia. See, this bluish color is there, no? Conidias. Conidia are formed externally on hyphae or more commonly on morphologically differentiated conidiophores. Sporangiophores are and conidiophores are sometimes characteristically branched and serve as a useful taxonomic creations are there. These conidia externally branched, unbranched hyphae tip termed as the conidiophore. The conidia may be formed single in a chain. The conidial chains may be basipetal, acropetal in succession. These conidias may be uninucleate or multinucleate. The lateral more common in members of Deuteromycotina. The sporangiospores may be motile or non-motile, and the motile sporangia are called zoospores. Motile sporangia is called the zoospores. This one. Motile sporangia or zoospores are provided with one or two flagella. Listen careful. This vital is the one zoospores two found into the flagella. How many? One or two flagella. The non-motile sporangia spores are called aplanospore. They lack flagella. Zoospores are this one. Zoospores commonly found in lower fungi, Pythium, Sporolingia, naked spores, of which after swarming insects create a cell wall, germinate from germ tube, thallus. They are equipped with one or two flagellas are there. These zoospores are naked. All zoospores are the naked spore lacking cell wall, which after swimming period, swarming period, Ensites. Ensites meaning here swarming period ensite. Ensite meaning that is create a wall and germinate to form a germ tube. Ensite meaning I'll explain. That is the name meaning of ensite. Create a wall and germinate to form a germ tube that develop into the thallus. These thallus, dermatoid thallus. These the flagella here two flagellas are there. No, see you can observe two flagellas. The flagellas are of two types. One is the tensile type, another one is a whiplash type. Tensile type, you can observe the tensile. This is the pitioscagenesil and whiplash is the filamentogenisil. Tensile type of flagella have a large number of air-like outgrowth. Tensile one, large number of outgrowth is present. A large number of outgrowth, and in this tensile type, this is the hair-like growth is also there. The whiplash flagella rhizome whip having bordered and ridged basal portion and a thinner, small, flexible and digital region. The flagella originate from a kernel-like granules are present. These granules are name is called blepharoplast. That name is called blepharoplast. Lying in the cytoplasm, lying in the close to the plasma membrane. The blepharoplast is contact to the nucellus by strand is called rhizoplast. That name is a cross section of the shaft of the flagellum shows in electron microscope that arrangement 9+2 arrangements are present in that one. This is a flagella. Next one, conidia. Conidia are two main types are there. Thallospores are the example arthospore, chlamydospore. Arthospore one, chlamydospore. The second one, conidiospore or true conidia. Here, thallospores are formed by transformation of pre-existing cell of the thallus and are detached by decay of the hyphae or disarticulation of the cells. The conidiospores, on the other hand, are formed as a new structure on the thallus and are easily detachable. This is the conidia. Now, the later type, the conidia may be sometimes uninucleate or multinucleate. The later type is most common the member Deuteromycotina. Coming to the Odia. Odia, see. Odia, another name, arthrospores. Odia is another name is called arthrospore. This is the arthrospore. Synonyms is called the Odia. Odia are formed by close septation of the distal end of the hyphae in a basipitular succession, producing. And what is a basipitical succession in Odia? That is the producing from apex to the base of the hyphae. Both here exogenous arthrosporous, exogenous and endogenous arthrospore are known, that is Odia. The exogenous arthrospore, Odia is the exogenous arthrospore separate by splitting of the transfer septate. You can observe to the C. In the picture, this is a Odia. Okay. This one is a Odia. Separate the transfer septa. The in endogenous formation of arthrospore, transfer septa are laid down as in the exogenous arthrospore, and neighboring or alternate cell develop a thin wall. The intermediate cell lost their contents entirely, and the thin lateral walls beat to free the arthrospores. Some pieces of hyphal remains remains attached at both end of the arthrospore. This is the Odia, or another name arthrospores. You can observe the picture. Next one, excuse me. Next one, chlamydospores. Another one, yellow color picture you can observe, chlamydospore. They are thick-walled resistant spore. They are thick-walled resistant spore formed by terminal or intercalary cell of the hyphae and are released after death of the hyphae. Occasionally, chlamydospores may be formed from cell of other kinds of spore, but best example, Fusarium. The chlamydospores are formed on the macroconidia. In the formation of chlamydospores, the cell round off, usually enlarge and develop a thick, often colored wall. The cytoplasm becomes dense with enough reserve food for consumption during unfavorable environmental condition. The chlamydospores are resistant to lysis by chemicals produced by other soil organisms. It has been reported by Cook and Sanders scientists in 1965 that chlamydospores formed on the macroconidia of Fusarium survive under condition in which conidia and the hyphae are quickly lysed. This is the chlamydomonas sexual spores. Coming to the ascospores or conidiospores, ascospores, basidiospores, see uridospores, and conidiospores are there. First one, ascospores or conidiospores, both are the same. True conidia are most common. You can observe to the Ascomycotina fungi and fungi imperfecti. In lower fungi, some genera from two conidiospore, while in some other, the example albugo and downy mildew, downy mildew, the sporangia in dry weather behave like conidia and germinate directly through a germ tube rather than forming zoospores. Such fungi are considered transitional forms between aquatic and terrestrial fungi, showing the possible evolution of conidia from the sporangia conidia, which may be uni or multi nucleate display by bizarre shape and are rarely thrilling to look at by nature or their beauty and color the conidia. These conidiophores may be free or arranged to form compound sporophore, and that is called the cinema, sporochidia. The conidiophore large number fungi are born inside structure that is a fruiting body, which may be saucer cup shaped acerbilly or flask-shaped globular pycnidia opened by the ostial. Detail about these structure will be studied in chapter Ascomycostina and Deuteromycotina, where the conidia reached their zenith. And in this one, the conidia, uridospores, very thick wall, red color, summer spores is the uridio. And you can observe to the rust disease produced usually on the leaves, surface or stem of the grasses. Basidiospores, they spore produced by Basidiomycetes fungi. They typical each contain on haploid nuclei. That is the product of meiosis, and they are produced by specialized fungal cells is called the basidio. In Fuchsinia, in the class of Basidiomycotina, different types of fungi there. These are things are you can study to the detail about to the Ascomycotina and Deuteromycotina time. I'll explain to the total different spores are conidiospores, pycniospores, aciospores, teletospores, basidiospores. These all spores you can study in the type study time. Okay. This is the asexual reproduction is totally complete. Okay. Now we can start from the sexual reproduction. See, here in fungi, sexual reproduction involves three principal events. How many principal events are there? Three principal. Plasmogamy, first principle. What is the first one? Plasmogamy. The second one, karyogamy. The second one is the karyogamy and meiosis. These three you can observe from the type studies of the sexual reproduction. And occurring in a cycle manner, plasmogamy here. First one is the, these are the sexual reproduction. First sexual reproduction meaning you can observe the plasmogamy is the fusion of two sexual cells, that a process which result in coming together of compatible. The sexual differentiated nuclei in a single protoplast in preparation for nuclear fusion. In next one is the karyogamy. Karyogamy meaning fusion of nuclei. Karyogamy meaning fusion of nuclei result in the production of diploid zygote nucleus. Production of diploid nucleus. Meiosis promotes genetic recombination through random genetic recombination through random assault of chromosomes or crossing over, resulting in the production of genetically different individuals. Next one, the sexual cycle of different fungi may vary due to variation in timings of the above event. In many Ascomycotina and most Basidiomycotina, the plasmogamy and karyogamy, they are separated in the space, the time. The compatibly nuclei do not undergo karyogamy immediately after plasmogamy, but remain paired and called a dikaryon. The dikaryon may persist for a varying period by consecutive division of two nuclei. What are the two nuclei are there? This nuclear phase in the life cycle is called dikaryof. And some fungi, next one, some fungi produced morphological distinct male and female sex organs, while in other, for example, mucor, these are mucor in this one. These are indistinguishable. Most of the Basidiomycotina do not produce any morphological sex organs, and the sexual function is taken up by cells of the stomatic hyphae. The sex organs of fungi are called gametangia. These may be from sex cells, the male and female gamete. Male gametangia called antheridium. Female gametangium are called oogonium. The motile male gamete is called anthrozoid. And the female gamete contained inside in the oogonia as a differentiated nucleated mass. The egg or oosphere. Morphologically similar gametangia and gametes are called isogametes, respectively. Both are the gametangias. Coming to the next one, here in the sexual reproduction, different are there. One is the plasmogamy, isogamy, planogenetic couple. These are all there. First one is the palanogametic population. This is the very simplest type. There are 5 basic modes of union of sexual elements which bring compatible nuclei in juxtact position. These mode of sexual fusions are often referred to as method sexual reproduction. One is the planogametic copulation. First one, A1, you can observe that the A is the planogametic copulation. This involves the fusion of two naked free free gamete, one or both of which may be motile. One or which may be planogamy, which may be motile. Motile gametes are called planogametes. Depending on the size and motility of the fusing gametes, there are three types of planogametic population. One is isogamy, second one anisogamy, and third one is the heterogamy. When the two fusing gametes are of the same shape and size, they are called isogamy. See, in the isogamy, fusing gametes morphologically similar, motile, but physiologically dissimilar. These gametes are produced different parent. Example is syncritum. Second one, this is the isogamy. Next one in heterogamy. Heterogamy involves the fusion of a motile. See, heterogamy involves a fusion of involves the fusion of a motile male gamete with a non-motile female gamete. Motile male gamete with a non-motile female gamete whose pair, see, you can observe the picture, whose pair contained in an oogonium. The motile gamete, the motile gamete, the anthrozoid enter into the oogonium and fertilize the egg or oosphere. Heterogamy occurs only one genus is there, that is a monobelfaris. Allomyces oogamy, and syncretrium is the best example. And next one, isogamy, anisogamy, which occurs only in one genus, Allomyces criteria, and two planogametes are morphologically similar but different in size. That is anisogamy. Coming to the gametangial contact. Next one is the gametangial contact. In this type of plasmogamy, the male gametes is usually represented by the nucleus contained inside the antheridium, while the female gamete is represented by the egg, contained in the oogonium. Sometimes both male and female gametangia contain a nucleus representing the gamete. When the two gametangia come in contact, the male gamete nuclei migrate into the oogonium, enter through a pore dissolved at the point of contact or through a fertilization tube, especially developed for the purpose by male gametangium. The two gametangia do not fuse and retain their identity. The oogonium undergoes post-copulation changes while the antheridium usually distinguished. For example, this gametangia, Phytoptera, Pythium, Albugo. And next one is the gametangial copulation. The entire contents, the entire contents of the two gametangia fuse and become entire, that one is a fuse and two gametangia fuse and become one. It occurs in two types. One is the directly fusion gametangia, directly fusion of gametangia as exemplified by Mucor, Diplodocus, Erminas, etc. Here the two gametangia fuse and become one cell, direct fusion. Migrate of entire protoplast of one gametangia into the other through a pore. The total entire gametangia act as a gamete. The recipient gametangium is called female, while the gametangium that empties its content is the antheridia. This is the best example, rhizopodium, and some other holocarpic critidas. Coming to the another one, spermatization. Spermatization. This occurs in some Ascomycotina. The best example, Neurospora, laboratory fungi use, and Basidiomycotina. Basidiomycotina is the see here, Fucsinia. Basidiomycotina is a Fucsinia. Here, minute conidia like male gamete. Minute conidia like male gamete called spermatia. That is called spermatium are produced on spermatiospores born externally on hyphae or inside cavities is called spermogonia. On contact with the female organ, the spermatia empty their contact through a pore. The female organ may be a female gametangium, specialized receptive hyphae, or even sometimes some stomatic hyphae. The spermatia are carried to the female organ through wind, water, insects, etc. The spermatia differ from conidia in being smaller and incapable to germinate and gives rise to a produced new thallus. This is the spermatization. Coming to the somatogamy. Somatogamy, you can observe the total pictures, okay? Somatogamy, fusion between undifferentiated vegetative cells or spores called stomatic copulation or somatogamy. In mycelial forms, anostomosis. Anostomosis meaning fusion between vegetative hyphae. Somatic copulation, somata in mycelium, anastomosis, fusion between vegetative hyphae. Phenomena common in higher fungi but absent in lower fungi. This method is followed by reciprocal nuclear migration, each mate fertilizing other. The compatible nuclei usually start a dikaryotic association for a indefinite period prior to karyogamy. Here, the karyogamy, after coming together, the two compatible nuclei may undergo fusion. That is a karyogamy. Immediate to gives rise to a diploid, to a nucleus. Or maybe start dikaryotic association extending from varying period in different fungi. The two nuclei evenly fuse to form asci or basidio. In Basidiomycotina, the dikaryophase is extended through dikaryotic spore like asiospores, euderiospores, teletospores. This is the total sporocarpus. Total fungi reproduction for the sexual reproduction and asexual reproduction, vegetative, sexual, asexual reproduction. See, this was picture, sexual reproduction of fungi. This one is the B, C, D, A, B, C, D. This is the A. This one is the isogamous B. This one is a C. This one is a D. Gametangia contact. C. The antheridia, oogonia. Female, male. Fertilization tube. This is a nucleus, oogonium. This is the ooplasm. Anthridium. This one is the anthridium. Fertilizing tube enter into the male, into the female nucleonium. And isogamous, see, male and female. See, this one also, male and female. Same heterogamy. Heterogamy, same male and female. Next, EFG, spermatangia. EFG. All are the spermatangium. See, hyphae, stomatic hyphae. You can observe bunch-like structures are there, no? These bunch-like structures are spermatium. See, this is the spermatiophore. Spermatiophore. Coming to the F, hyphae. This is the hyphae. Spermatium hyphae. This is ascogonium. This one ascogonium. This is a trichogony. Trichogony, spermatium and trichogony. CH part. This H, gametangia population. See, hyphae of matting to one is a negative and positive. Both are combined gametangium to after fertilization to formation of zygote. This is called zygospore. This is one zygospore. The final I1 somatogamy, hyphae of opposite mating type. See, this is the one hyphae of opposite mating, another one to fusion to form like this. These all are fungi reproduction by three methods we will discuss today class. The first one is vegetative. The second one is asexual reproduction. The third one is the sexual reproduction. Total, this one is completed. Another one now to enter into the classification of fungi. What is the taxonomy, nomenclature, and classification of fungi? Taxonomy, classification. Here, oh, it's very big. Uh, Taxonomic nomenclature and fungi classification of fungi. Here, organisms. Oh. Organisms are classified in a hierarchical series of groups. The science of classification is called taxonomy. What is the taxonomy of fungi? And now I'll explain. Now, organisms are classified in a hierarchical series of groups. The science of classification is called taxonomy. It is different forms nomenclature. The term systematic, which is often used as a synonym of taxonomy, is a broader concept which includes identification, nomenclature, and the practice of classification. Taxonomy is concerned with both identity and status. Thus, a taxonomist must decide not merely when what species a particular specimen should be assigned to, but also its relation with other organism. For discovering relationship, a knowledge about the organism is necessary. The relationship is searched on the basis of resemblance and difference. Depending on the criteria used for classification, taxonomy is of several kinds. The oldest and most widely used classification of plants and animals is based on morphological based on morphological character. It is called classical taxonomy. Although although morphology continues to be important, the modern trends in taxonomy emphasize upon biochemistry, biometry, cytology, ultrastructure, etc. Besides the search for better morphological criteria, thus we have biochemical taxonomy based on distribution of chemical compounds. Numerical taxonomy applies mathematical procedures. Cytotaxonomy based on characters of somatic chromosome, and experiential taxonomy based on the analysis of various patterns of variation. The success of taxonomic classification lies in different parts. One, keeping track of information about organisms. Second one, summarizing this information. And third one, predicting the characteristic of incompletely studied organism. Taxonomy is an investigative since science. Our concept about an organism changing as we gather more information about it. The taxonomic grouping recognized at any given time are indicative of our knowledge at that particular period. New taxa continuous being discovered. This necessary frequent adjustment in the hierarchy of the various taxa consisting of varieties, species, genera, families, and orders. These processes constitute taxonomy. Thus, taxonomy is ever changing, signifying increased knowledge about organism. Nomenclature, of the other hand, aims at suitable, unlike taxonomy. It is not an investigative science. It is merely giving name to an individual according to accepted norms of an International Code of Nomenclature, ICN. Correct names can be assigned only after the taxonomic status of organism has been decided. Nomenclature thus exchange with a change in the status of the organism in taxonomic ladder. And this announce the applied mycologist like plant pathologist, industrial mycologist, physiologist, genetics, but can't be helped. Species concept, based on the species concept, morphology, host specialization, both morphology and physiology, the same criteria cannot be applied to all groups of fungi. Characters used in classifying species of the one group. Example, Phytophthora may be of no use or even non-existence in another group. Example, Fusarium or Rhizovirium. Natural year and artificial classification. Natural classification attempts to place organism in an orderly arrangement on the basis of overall resemblance, preferably on genetic basis, classification based on one or few convenient characters to serve special purpose, that is the ace of identification, is called artificial classification. It may have no genetic significance. The natural classification reflect degrees of evolutionary relationship and therefore may be pathogenetic. The taxon is general term as taxonomic group, where it rank kingdom, division, class, order, family, genus, kingdom. Okay, this is the taxonomic classification and nomenclature classification. We will discuss about next week class. Okay, thank you. I'll stop my sharing.